Hello students. Let's begin with the big idea behind animal kingdom. Animals are not grouped randomly. They are classified by levels of organization, symmetry, body cavity, segmentation, and the presence or absence of a notochord. If you learn these 5 bases properly, the whole animal kingdom starts making sense as a structured progression, not a memorization exercise. The first universal rule is simple. Every animal in Animalia is multicellular. That sounds obvious, but it matters because the real distinction begins after that. Even though all are multicellular, they do not organize their cells in the same way. That difference in organization is the foundation of classification. Some animals stay at a loose cellular level, while others move into tissue, organ, and organ system levels. Animal evolution does not jump directly to complexity. It builds step by step. First comes the cellular level, then tissue, then organ, and finally, organ system. That staircase is the backbone of the chapter. Once you see this progression, the classification of phyla becomes much easier to remember because each group fits into a specific stage of organization. The cellular level is the simplest organization. Cells are present, but they remain as loose aggregates. They may share the same space and even divide labor to some extent, but they do not form true tissues or unified structures. Sponges are the classic example here. Here's a trap you need to be aware of. Division of labor by itself does not automatically mean tissue level. The arrangement has to be structurally unified for that. The tissue level is the next upgrade. Here, cells performing the same function are grouped into tissues. That means the cells are not just similar, they are organized into a coherent structural unit. Coelentrates are the standard example. A very important exam clue is the phrase same function. That phrase tells you the question is testing tissue level organization rather than just cell grouping. Here is one of the most missed points. A sponge may show different cells doing different jobs, but that does not make it a tissue level animal. Tissue level requires a rigid structural arrangement of cells doing the exact same job in an organized form. That is why cellular and tissue levels are not interchangeable. If you blur that distinction, statement-based questions can trap you very easily. The organ level is a bigger step. Now tissues group together to form distinct organs. Each organ is specialized for one specific physiological function. Platyhelminthes are a key example here. This is the first time you see a clear, visible anatomical part doing a dedicated job. So the shift from tissue to organ is not just about size. It is about specialization. The organ system level is the highest structural organization shown here. Different organs work together as a functional system, and each system is specialized for a larger physiological task. Annelids, arthropods, mollusks, echinoderms, and chordates are placed here. That means their internal complexity is not just about having organs, but about integrating those organs into systems. Digestive organization is a great way to understand systemic complexity. Some animals have an incomplete system, where a single opening serves as both mouth and anus. That means food enters and waste exits through the same opening. Platyhelminthes are the example here. Others have a complete system with separate mouth and anus. That design is more efficient and more advanced. So one opening versus two openings becomes an easy comparison point in classification. Circulatory organization follows the same logic. In an open circulatory system, blood is pumped out of the heart and directly bathes the tissues. In a closed circulatory system, blood stays within vessels like arteries, veins, and capillaries. Closed circulation gives more control and efficiency, which is why it appears in more advanced animal groups. This contrast is one of the cleanest ways to compare body systems across phyla. We will now discuss a quick diagnostic cheat code. Sponges belong to the cellular level. Coelentrates and ctenophores belong to the tissue level. Platyhelminthes belong to the organ level. Annelids to chordates belong to the organ system level. If you can map the phylum to its level of organization instantly, a large part of classification becomes much easier in objective questions. Body symmetry is another major classifier. Some animals are asymmetrical, which means no plane can divide the body into equal halves. Sponges are the classic example. Radial symmetry means the body can be divided into similar halves through multiple planes passing through the central axis. Coelentrates, ctenophores, and echinoderms show this. Bilateral symmetry means only one plane can divide the body into identical left and right halves. Annelids and arthropods are the key examples. Here's a trap you need to be aware of. Symmetry is about possible planes of division, not about appearance alone. Embryonic architecture adds another layer to classification. Diploblastic animals have 2 germ layers, while triploblastic animals have 3. Then comes the body cavity or coelom. A coelomate has a true body cavity lined by mesoderm. A pseudocoelomate has a cavity, but it is not fully lined by mesoderm. An acoelomate has no body cavity at all. But be careful here. Ascalminthes is the only pseudocoelomate group highlighted here. That point is often used directly in exam statements.
The final upgrades are segmentation and the notochord. Metamerism means the body is divided into repeating segments, internally and externally. Earthworm is the standard example. The notochord is a mesodermally derived rod-like structure formed during embryonic development on the dorsal side. This becomes the great divide in Animalia. If notochord is present, the animal belongs to chordata. If it is absent, it falls under non-chordates. That is why the notochord is one of the most important classification markers in the entire kingdom.